Hello, Internet. I'm Mike. Welcome to the INC Chalk Talk series. This is Pamela Reinagel, who will be giving a talk about control in the visual system.
Thanks. So it's really fun to talk about this project. I took advantage of the Chalk Talk name of the series as an invitation to talk about something that's new and that I haven't talked about or published on yet. And so this is a very early stages of a project where feedback would be extremely useful. So I'm going to start with some backstory. Actually, when I was first started thinking about the brain when I took courses in psychology in college, way many decades ago, and one of the things that I was particularly fascinated about.
I wonder if something horrible like suicide happened.
Yeah, I know.
Okay, I think there's a microphone.
Or an overdose.
Whoever just logged in,
Because they're being so closed-mouthed about it.
We can hear everything.
I don't have any.
there we go.
All good. Okay, I want to stand by.
Maybe I'll sit back here.
Okay, all right, so let me introduce this topic again. So the question that this talk is, pertains to, my interest in it goes back to when I first learned about patients, such as the patient that's described here, but that have been described all the way back to World War I era, that have damage to part of their brain that affects some visual capacities and not others. And in particular, this particular task shows a person who's holding a flat object in their hand, and they're being shown a slanted line, and they're being asked to report what angle that line is at by holding their object at the same angle. And a control subject, which is shown on the bottom, can do this with extremely high precision. Whereas this patient, DF, was completely unable to. They reported that they could not see the orientation of this line. And they could not, even if they were forced to guess, they could not guess its orientation. But in another variant, what might have seemed like the same task, they said, okay, take that flat object, pretend it's an envelope and stick it in the slot. And they reached out and hit that angle with extremely high precision. And so this was one of the patients that was importantly led to this idea of the parallel visual stream or dual visual stream hypothesis, that said that the visual information that's coming into the brain from the retina, passes through the thalamus to primary visual cortex, flows after that in two parallel streams in the primate visual system at least. One that goes to the inferior temporal cortex, the ventral stream, which is responsible for encoding form, color, object recognition, these types of features. And another that flows toward the dorsal stream that's responsible for encoding motion and maybe the location of objects and specifically the control of actions such as reaching, gripping, orienting objects like was in that task I just showed you. So that a patient who had damage in inferior temporal cortex might not be able to perceive or report the form of an object, but might still be able to take motor actions that are guided by the visually perceived, the visually detected form of that object. So it's a super fascinating example. But then there are,
Yeah, sorry, just a quick question. So what can they see exactly then?
you know, I didn't recently review the patient's self-reports on this patient. So all I know is that they could report visual perception of color, but not of shape.
Okay, but then their motor action still reflected some understanding of something they consciously were not aware of.
Exactly.
Okay.
Exactly. And an even more surprising case of this are patients who have lesions in their primary visual cortex. And this is a picture of one such patient whose face has been covered for you. That's not their blindfold. This is a patient who has what's called blind sight, meaning that if you ask them what they see, they will tell you they see nothing. They are blind. And yet, and so normally the interview would end there in the neurologist's office, but the neurologist then thought to say, okay, but am I holding up my left hand or my right hand? And they say, what, I can't see. They get really mad. I say, okay, doesn't matter, just guess. And they can sometimes guess correctly. And in particular, they can do certain tasks, in such as the one that's being shown here, which is walk through a room avoiding objects in their pathway that they aren't aware of having seen or avoided. So a lot of people have been interested in this phenomenon from the point of view of consciousness, right? We have information from the eye reaching the brain. We have no visual awareness of it, and yet it can still guide some of our behaviors. Obviously, it's still reaching parts of our brain. And this one's a little bit more mysterious because the lesion is in the primary visual cortex, which is supposed to be where all the information arrives in the brain, right? So it should be cutting off both the dorsal and the ventral stream. And so when cases like this were reported, they were hotly debated. And I'm going to move to a drawing here from Ramona Cajal of what we've been calling the primary visual system for quite some time now. We have the retina in the bottom left, capturing light and sending information down the optic nerve to the thalamus. The nucleus in the thalamus is called the lateral geniculate nucleus, or LGN. And those neurons send information to the primary visual cortex up there on the upper right, which then sends information to other cortical areas, what in the mouse literature people now call higher visual areas, or HVA, such as the dorsal and ventral streams. And so some of the hypotheses explaining blind sight in patients with V1 damage was that maybe part of V1 was really spared, or maybe the LGN was sprouting new connections to other parts of the cortex, or weak projections of LGN directly to higher visual cortex could be accounting for the behaviors, and all of these things can happen. But the rest of Cajal's diagram points to another possible explanation, which is that the retina, besides sending axons directly to the LGN and the thalamus, also sends its axons to another image-forming nucleus called the superior colliculus. So this is another subcortical target that receives a retinotopically organized retinal projection. And the superior colliculus is the homologue of what in a frog or a lizard would be called the tectum, and that's the visual structure that mediates all visual behaviors of those creatures, because they don't have a cortex. And so to me, it seemed like a very plausible hypothesis that our capacity to carry out some visual behaviors, especially direct motor behaviors with visual information, in spite of not having any visual perception, conscious perception of it, might be us experiencing what it's like to be a visually behaving lizard, right? Yes.
That's true of fish too.
Yes, and yeah, and even lampreys.
Okay.
So yeah, it goes through vertebrates. It's a little bit more murky with birds because they don't have what we would recognize as a mammalian cortex, but it's argued that they have a similarly organized neural circuit. So they're sort of in between. But every vertebrate, I believe, has a homologue of the superior colliculus, and it also receives a structured, retinotopically structured projection from the retina, which means that the information is laid out in space across the tissue according to where it is where the light came from in space in the world. Okay, so the other possible explanation for vision without V1, visual behavior without V1, is that the information from the retina is passing through the superior colliculus. It goes through another thalamic nucleus called the pulvinar and makes its way directly to some higher visual areas, and therefore could underlie visually guided behavior without requiring V1. And apparently, this pathway is not accompanied by conscious perception of the visual. The people are conscious, right, but they're not conscious of a visual awareness. So I've always been really interested in this problem, and now I'm going to make a hard transition to how I came into the system many years later, when my lab decided to set up a weight behaving rodent preparation for studying vision. And this was back in the day when most of the vision science world insisted that mice and rats were nocturnal and probably blind, and the majority of the research was being moving in the direction of monkeys. But we felt there would be some experiments better done in the rodent preparation, and so we set up. We looked at a bunch of different rodents. I'm not going to get into that story now, but we ended up picking pigmented rats as our model system, and we built this thing we call the Ratrix. where every rat has attached to its own cage, a little operant chamber with a computer screen. They each have their own computer that is running some task, some visual task, and they have to interact using lick ports to perform these visual tasks in order to get water. And this is the only way they get water ever. So they live in this cage, they have this task available at all times, and it's absolutely essential for survival. And so if you were to come into one of our vivarium rooms, it would, any part of it would look sort of like this, a bunch of rats in their cages doing tasks. So I'm just gonna quickly introduce some of the tasks. So this rat here on the upper left is doing an orientation discrimination task. So it has to report, it has to go to the side where it thinks the stripes are horizontal to get water. And we're varying things like the contrast or spatial frequency to probe their visual acuity in that particular task. In the upper right, we have an animal that's learning to do a object image discrimination. So this animal has to mark where the statue is. Yeah, checkerboard. On the left, we have we got motion discrimination. We have to respond on the slide that it thinks underneath. Left, right, is the first task, demonstrator has to do a primary discrimination task. Here we're not at seas, we're in a faint target in the center of the screen while exploring similar flanking stimuli. That allowed to stimuli.
Oh, thank you.
Yeah, I know, that's a lot.
So close.
Okay.
That's so hard.
There was definitely some background noise that the rats would not be hearing, but the little beeps they do hear. And a lot of labs build like soundproof chambers around every animal so that they won't distract each other. Our animals have to function in a very noisy, busy environment. So, so they would be hearing all the other animals beeping, and they figured it out. Anyway, so this is the story of one rat that was unusually talented and that it learned to perform a number of these tasks. Most rats learn only one or two. And so this rat, this is the training history. There's a little gap in the training history indicated by the hatch mark. That's for one task that the rat failed to learn. But the tasks after they were learned to 80% criterion were indicated by these different colors. One is report the location of an object on the screen. So a picture of a statue shows up on the screen and you have to tell me where it is. Blue is report the location of the statue and not the space shuttle. So it's discriminating between those two objects. The green and cyan are two different versions of the random dot motion task. So essentially the same task with just different parameters. The magenta symbols are report the location of a gradient patch, and the black symbols are report the orientation, so go to the horizontal and not the vertical, for example. And so this animal mastered all of these tasks, and so we performed a lesion of V1 in this animal, and after recovery we asked what how that affected its behavior. Would it, first of all, at that time, it wasn't formally known whether V1 would even be necessary for any of these behaviors, right? If someone can post an envelope to a slot without V1, why shouldn't they be able to go to an oriented grading without V1? Motion is one of the things that is supposed to be computed in the ventral stream, and it's possible that mice could do motion without V1. And so the paper that we published on this, which is cited here at the bottom, you know, our title was actually V1 is required for several of these tasks. But it is not required for others, which is the part of the story I'm talking about today. So after the lesion, the animal had a profound deficit. It was reduced to 0.5 or chance on motion discrimination, orientation discrimination, and object discrimination tasks. And it was tested for almost a month and it never recovered at all. So it was very complete and stable deficit. On the other hand, it returned to its pre-lesion level and continued to improve and reach ceiling on the two tasks that involved reporting the location of an object. So oriented behavior running over towards the direction where an salient target was displayed was completely spared. And so, while I kind of buried the lead in this paper, when I reported this, this really excited me at the time because it means we now have a rat model of line sight, a behavior that does not require V1, whereas V1 is required for many other visual behaviors, which supports at least the conjecture that the mouse's visual experience might be like human patients with blind sight, that their cortically supported behaviors might be consciously perceived, and their V1 independent behaviors might be without conscious visual perception. Okay, so this is how I'm imagining this might work in a little bit more detail. So this is a super cartoon diagram of the visual system. So we have the right and left retinas indicated by these curved lines that I'm using color to indicate which side of the world the images are coming from. And I'm just going to talk about images that are coming from the left or right side, so leaving out the binocular field entirely. Just simplifies the story. So any stimulus is on the right side is going to be represented only in the left superior colliculus. And the left superior colliculus has motor output that commands locomotion to the right. And so a stimulus on the right could reach the colliculus and elicit a behavioral output in the right, to the right side of space. And the opposite for stimuli on the left would go to the right colliculus, which sends motor outputs to move towards the left side. And then the two colliculi are connected between hemispheres of the brain, and they mutually inhibit. So if you have only one stimulus, generally only one colliculus will be activated and the animal will respond in that direction. But if there are more than one stimulus available at the same time, then this mutual inhibition circuit implements a winner-take-all algorithm that assures that the animal just picks one or the other as the output target of behavior, instead of some kind of vector average, which would put it where nothing is. Right? Yeah.
This may be a naive question. I was thinking previous slide. Is it possible that you have V1 does some processing and then discrimination is done by another module connected to V1? So
yes, I'm not claiming that these discriminations occur in V1.
okay,
But just that they're dependent on, they're dependent on information passing through V1. I would imagine that these could be discriminated in different visual areas. In fact, our simplest hypothesis would be that object discrimination would be like the equivalent of the infratemporal lobe discriminations in humans. Motion discrimination might be like the equivalent of area MT in a monkey, and orientation discrimination might be the only one that's computed in V1.
okay,
But we haven't shown any of that.
okay.
We've just shown that V1 is required.
So it's so also then you kind of there's evidence that superior colliculus is not, if that's true, is not connected to any of those modules or contexts.
I have not ruled that out.
Okay.
Yeah. So
Also, how total was the V1 lesion?
this one was quite complete, but I have to say we had a number of other lesions that were partial, and even though they were substantial, they had more partial or variable deficits and spared behaviors. It's very hard to interpret that because the animals are freely behaving. So if they've just got a little spared part of their visual field, they might learn to like put the stimulus into that part of their visual field to make the decision. And so this one is the only one we published because it had a very complete lesion and a very clear association. It's possible that we took out a little bit more than V1, and so I wouldn't say that we really nailed down the boundaries of which exact place in the cortex is required for this. I'm trying to simplify the story a little bit. So, yeah.
So I just want to double check, those two symmetricals on the left, those are retinas?
Yeah.
Okay. And that's just the left and the right now?
Yeah, so this is the left eye, right eye.
Okay.
And I'm just showing that bare minimum skeleton of a diagram here. So the idea is that the colliculus enables animals to, the hypothesis is that the colliculus enables animals to approach to have a motor output in the direction of a single salient target through this very simple kind of circuit that I'm showing here. But this is not selected for things like orientation or image identity. And then area V1 is required, and maybe other parts of cortex, but definitely area as well. With all the caveats I just said, the part that was lesioned in that one rat was required in that one rat for discriminating images or orientation, for example. But interestingly, we know from other people's work and other sensory modalities as well, that even when you're doing a task that requires cortex for the discrimination, the motor layer of colliculus is still perfectly correlated with the oriented motor behavior. So the colliculus motor layers are essential for the animal orienting its body in space towards some behavioral target. So that means that when we're making a discrimination using cortex, that information has to make its way back to the colliculus. And there's many possible pathways, direct and indirect, but I just drew a couple of the more direct ones, which is that when the animal is doing something like orientation discrimination, the discrimination could be happening in V1, and then V1 sends news of what happened back to the colliculus. And there are two pathways I highlighted here that are my current working hypothesis for pathways. One is that V1 sends direct excitatory projections to the colliculus in a way that's spatially aligned to the visual map in the colliculus. So, so if V1 detected the target orientation, it could excite that part of the collicular map and help the colliculus win that winner-take-all competition. Another possible pathway is that V1 also sends projections to the striatum, which then sends inhibition to the colliculus. So it's possible that V1 detecting that it's the wrong pattern could be indirectly inhibiting the colliculus that would otherwise cause the collicular response to behave in the direction of that target. So some kind of feedback from cortex into colliculus is biasing the outcome of this competition. And so that's sort of a different way of thinking about what's happening when animals are making decisions in this kind of task. Yeah.
One question, in this case, go first, I understand that sometimes in the eyes, the retina leave the optical nerve and divide into in the vertical and the right and the left.
Yeah.
But in your diagram or your circuit only mark in one direction,
Yes.
or
Yeah.
left or
Yes, so the reason for that is that if you look at the colors on the little semicircles here, there's a teeny bit at the edge that is also receiving input from the other side of space in each retina. So that's the binocular field in front of the animal's face. Those are the only axons that cross over.
yeah, but
And
in
I
this,
didn't show them because I'm only going to show stimuli on the sides, so that we keep them isolated. It's just to make the picture simpler. There would be an equivalent sort of picture could be drawn for what's happening in binocular space, or if I put two targets both on one side. But I'm just for the sake of trying to make a very simple cartoon, I just chosen to design my hypothetical experiment so that we're only putting things at the part of the visual field that exclusively projects to the other colliculus.
but the circuit is very similar to the humans. The rat is similar.
Yes, it is, but the humans have a huge fraction of our visual field is binocular, and for the mouse, it's very little, because their eyes are lateral.
But the distribution of the circuits are.
They're very thin.
Okay.
The cortex, there's a lot of differences. There are a lot of differences, but that part of the circuitry is the same. Okay, so just to walk through, this almost toy level model, just to give you the intuition of how I'm thinking about how these behaviors work. That V1 is somehow biasing the outcome of competition between the two colliculi. Could be how pattern discrimination impacts the behavioral output of the animal in this task. And when we lesion V1, then the outcome reverts to whatever the colliculus would have chosen to do. In other words, without respect to pattern. So discrimination tasks, the way we design them in the lab, usually match the salience of the two patterns that we're trying to discriminate. So we'll take two gratings that have the same contrast or spatial frequency, for example, and only change the orientation if we want to probe orientation discrimination. Or if we have these two photographs, we match the total luminance, total area, and total root mean squared contrast, so that we try to match the salience as well as we could, so that they would be discriminating the patterns. And the consequence of this is that, as far as the colliculus is concerned, these two stimuli are designed to be equivalent. So when you take V1 offline, the reason you go to chance or 50-50 is because the two colliculi are getting the same intensity of salience. And therefore the outcome of the winner take all competition is just chance, it's just random. Whichever one randomly happens to get greater than the other one will eventually take over and control the behavior. But that's not the way it works in the real world, right? In the real world, if we are trying to find, let's say, the oriented target, we're trying to find Waldo in a picture, looking for a particular pattern, that may not be the most salient stimulus, right? The stimulus that we should be approaching on the basis of this pattern might be the less salient stimulus. And when that happens, we would create a conflict between colliculus's ability to guide behavior, which we can see colliculus is totally competent to guide your oriented behavior. And it wants you to go to the salient target, whereas the cortex, which is doing the pattern discrimination, would be dictating the opposite behavioral action. And so this poses to me a really interesting and tractable model for thinking about what happens when maybe our more evolutionary ancient circuitry in our brain has to coexist with our modern cortex when they disagree, like they have to somehow wrest control. One or the other of them has to win control of behavior. And so I think it's very interesting to understand like what determines the outcome of that contest. So the question, just to put it in the space of this drawing, is what if we had conflicting salience and pattern cues so that the S plus or rewarded pattern stimulus was the smaller or less salient, lower contrast stimulus, and the wrong pattern stimulus, S minus, was the larger or more salient stimulus, then we would have the signal from the retina to the colliculus strongly dictating that we should be responding to this more salient pattern. Whereas, which in the absence of E1 would mean there'd be a strong bias to respond to the more salient target instead of the less salient target. But then if we tell the animal they're only going to be rewarded if they go to the correct orientation, the only way the animal can learn that task would be if B1 is strong enough to choose the stimulus that has the right orientation and send a strong enough signal to its colliculus to allow it to win this competition. So it has to override this existing and probably earlier strong collicular direct retinal pathway. So we have a retinal signal telling this colliculus to win and a cortical signal telling this colliculus to win. And then there's a dynamical battle until somebody wins. So if this is happening, right, that's pretty impressive that this cortical input would be able to accomplish that. And it does suggest that in tasks where the salience is not matched, we should expect that when the conflicts come, when the cues come in conflict, it would probably induce errors. Right, when animals make mistakes, they might be more likely to choose a salient target in spite of the fact that it's the wrong target, wrong pattern. So to test this, we need to have a task in which the salience of the stimuli are not coupled to the pattern, such as this image discrimination task that's invariant to size and incidentally also invariant to rotation. So in the interest of convincing ourselves that the animals really could tell a statue from a space shuttle and weren't just latching on to some local feature or subjective difference in salience, we had built into this task that the sizes of the two images were varying independently over a twofold range, and also their rotations were varying independently over some range. So the animals that learned this task had to learn to go to the statue even when it was the smaller or less salient stimulus. This is an example of one rat's learning curve as a function of days that it was put on this task. And this is a really industrious rat. He did 1,200 trials a day. And so by the end of the first day, had already exceeded 70% correct on this task in spite of this challenge. And over the course of, I guess it was 7 days, was doing something like 80% correct on this task. And so this gives us the possibility of testing our idea that when the colliculus and the V1 were in conflict in that subset of trials, the animal would be more likely to make errors in favor of the more salient target. So I'm going to take a second to walk through this because this is the main new result that I'm talking about today. I'm showing you a lot on these plots. There's just two rats, similar plots. I'll just use one for illustration. Two tasks. The first task is just tell me where the statue is. And the statue, you just shine it either on the right or left side, and you go to the side you think the statue is. And the animals are almost 100% correct on that task, and those are shown by these horizontal lines. And the horizontal axis is the size of the distractor image, and since there is no distractor image, it is not relevant to that task. And the colors indicate the size, it was continuously varied, but I just binned it into the smallest, medium, and largest sizes. And so red is small, made more errors when it was small, a little bit more accurate when it was medium size, and the most accurate when it was big, but pretty close to 100% correct for all three sizes for locating the object in space. And then we do this task over here where we introduce a distractor. The size is also varying independently. And so now we've got 9 conditions, right? Small, medium, or large target indicated by the 3 colors, and small, medium, or large distractors indicated on the x-axis. So if we start with the small target size, so this is the most difficult to detect, the red line there on the bottom, just introducing a small distractor made the task much harder, right? It made a lot more errors than when there was no distractor. And the bigger the distractor gets, the bigger the space shuttle becomes in this image pair task, the more errors the animal made, right? Which is consistent with the idea that the more salient the distracting stimulus is, the closer the contest between the 2 colliculi, and the harder it is for V1 to overcome that in that competition. When the target is very large, the animals do much better, but and the small distractors, hardly impair performance. But again, the largest distractor is able to significantly impair, which is consistent with what we'd expect. Yeah.
Sorry, John, just want to check again. So what is the difference between the straight lines and the ones that are actually going downward? Is it that the rat with straight lines is not, they don't have a lesion in V1?
No, there's no lesions at all. This rat is doing the task on the left. Only one object is present, and it just has to go to it. So there is no distractor in that task. So that's just detect and approach a target. So that would be what I am purely collicular task. And then the slanted lines are the V1 dependent task, which is go to the object because of its pattern, regardless of its salience.
And the spaceship will grow.
And the spaceship, they can be on right or left with equal chance, and their sizes are varied independently. So, yeah.
I mean, you don't have to do this experiment, but if you were to just give the distractor and change the size, you would still choose it, you think? Or
I'm not sure I understand what you're suggesting. Oh, the distractor alone?
yeah.
Oh, I don't know. So one of the, this has more to do with animal training, but one of the training principles we follow is that we never reward a behavior that the animal will later be required not to do.
Okay.
But if we did a probe trial with just the distractor, I would guess that it would go to it some of the time.
Okay.
And if it's mastered this task, it might be able to inhibit that to some degree.
Okay. Well, I was just wondering if, like, salience, since you said salience is also
Exactly.
behavior, so.
Exactly. So if I didn't do a diagram of that, but we could set that up. If we just did the distractor alone, we would expect a strong salience with Q to approach the distractor.
Okay,
And then it would be a big, huge job for V1 to overcome that and say, no, it's the wrong pattern. Don't go there. And I'm not sure, I think it depends a little bit on the rat and how much a penalty or punishment we give them for wrong answers, like
Right.
whether they would learn that. That'd be good to model and to show. So I just wanted to, because these diagrams take a long time to get used to reading, I just wanted to point out that if you just look at this set of circled points, that would be your standard discrimination task, meaning that the size or salience of the distractor and the target are matched. Right, so they're both small, they're both medium, or they're both large. And so if we were to do a typical or image discrimination task where we match the salience, we would see that, when both of them are larger, the tasks get easier for that rat to tell which is which. And everything up and to the left are cases where the target, the correct answer, is also more salient, and that makes it easier to choose. And everything on the bottom right is where the distractor is more salient, and that makes it more difficult to choose. So, and on the right is just another rat with very similar data.
What is the difference between rat one and rat two?
They are two different individuals, that's all.
But that's it?
They're just two animals.
That is
Just
what the
replication.
results of difference.
No, they're just two of the animals.
Oh, okay.
And they're, the first one I showed you was the third one. So, I think I'm showing that on the next slide. Yeah, so the first animal I showed you, the training course of, there's that animal graph on the right, so it's a similar story for that rat.
The same age, same learning process, everything.
So it is possible that the way the animals learn the task will matter. We haven't done a controlled study of that, but I would like to. I am trying to convince NIH that they should pay for that study. But in the meantime, and I think I'm gonna skip over this next point because of time, but this was just coming up with a way that we could measure this effect in small numbers of trials, because we don't always have 6,000 trials to analyze. I want to get back to how we imagine this works and what we're doing with modeling and planning for experiments. So here's a picture of a mouse head and, vaguely where its superior colliculus is, illustrating to you that there's a visual space in the world is mapped onto this two-dimensional map in the colliculus, the retinotopic map, and that retinal inputs come to the superficial layer of the colliculus. So this is just a side view of the colliculus, so you can see its laminar structure. So retinal input comes in here on the top layer and is organized according to space, and I'm claiming that projection is carrying mainly salience information. In the deepest layer is the motor layer, and that contains an aligned motor map of space as sort of an affordance map of behavior, where you can move and to get something you want. And then all the processing that's happening in between these two layers is informed by inputs from lots of other structures. So they're very integrated. It gets input from other sensory modalities, including V1. So V1 sends its map aligned inputs to this layer here. This diagram has more details that I want to get into, but it shows that a little bit better. So here's a retina and an object on that retina, and showing that the retina sends its projections to the superficial layer, organized. So the blue ones are near the center of the scene, are showing up at this end of the map, and the orange ones, which are for the periphery, are showing up at the opposite end, and showing that the map in V1 sends its projections to the layer below, lined up in space with the same part of the image. These little green blobs here are for those ones that cross over that you asked about, and I'm just avoiding thinking about them by putting my stimuli over here. And this is just illustrating, for example, somatic sensory cortex, but also auditory and other sensory inputs. And depending on the species of vertebrate, they have different sensory modalities, and they pretty much all send their inputs in such a way that the part of space that they represent is lined up to that place in the visual map of the retinal that's established by the retina. So I think of this as sort of a spatial map of behavioral affordance that's set up by the retina, informed by all these other inputs. And so we have the simplest possible computational model getting started to simulate this idea. So I've had this picture in my head for a long time, and it's just a hand-wavy argument. So I decided I better just write it down and implement it, and I'm gonna try and do it as simply as I possibly can. So for now, I'm encoding the stimuli with just two variables, a binary variable, are you the correct or incorrect pattern, and which I encode by design, and then a salience variable, which is scalar, and just says how salient is the image. And those are impinging, not really modeling the retina in any way, but just modeling the bit of a pathway that arrives in a colliculus with some weight, the weight between the retina and the colliculus. And for now, I'm modeling that those weights are the same for both hemispheres. And then there's some weight of this competitive inhibition, of this mutual inhibition, the intercollicular inhibitory weight. And then we have a weight of the whatever part of cortex is detecting the pattern, sending its input to the colliculi. And for now, I've only modeled the pathway that's a direct excitatory pathway from V1 to the colliculus. I mentioned there's another disynaptic inhibitory pathway, but but I haven't incorporated that into the model yet because I'm still working out how much we can compute with only this part. And then we have some noise terms and bias terms. And so it's a pretty low parameter model.
And all the circuits, the frequency of the spikes are the same?
There's no individual units in this model. So there's one unit for this colliculus, one unit for this colliculus, no spikes.
All spikes?
No spikes, just analog values.
Oh,
This is a very reduced model.
okay.
You know, my philosophy about models, which I know varies, you know, you can make them as realistic as possible. And that has some advantages. And the approach that I tend to take is make them as simple as possible and wait until I hit the limit of what I cannot explain. And then if I can't explain it without putting spikes in, then I'll put spikes in. And so I'm not sure yet that this model doesn't explain everything yet, but I'm not sure I pushed it as hard as I can. So that's why I haven't added that. So I'm just going to show a little bit of, if you look at how the collicular activity evolves over time in this model, on the top, it doesn't really matter because they're individual trials. You can only tell something from the behavior over a lot of trials. So it's the differences of conditions aren't so important as just to show that in every trial, the two colliculi begin with some initial activity, which doesn't have to be matched. There could be a bias. And then as a result, over many iterations of integrating the direct retinal input and the input from V1 and the interhemispheric inhibition, the activity of the two colliculi evolve and generally diverge because of the mutual inhibition. And so one or the other hits arbitrary threshold parameter that I set that I'm modeling as determining the decision. This much of a model is enough to account for some features. So this is the behavioral performance of that model. In the red line is the task that's just go to the one stimulus. The blue line is go to the statue, ignore the space shuttle task. And it, like real animals, that's a harder task. And so it's the curve as a function of contrast is shifted. And there's a couple of controls. If both of the stimuli are the correct pattern, then the animal should choose either one at chance. If both of them are the wrong pattern, they should choose either one at chance, but both of those are at 50-50 as you would expect. And if you lesion V1 in this incredibly limited model, then going to the correct target is preserved. Discriminating between the patterns is gone. And if you pivot this invariant contrast task that I just showed you, you get a pattern that looks very much like what we saw in the rats, which is that more errors are made when the distractor is more salient. And you can even get to the point if the distractor is much more salient than the correct image, that you can be systematically below chance because you're systematically choosing the wrong one. Yeah.
Maybe I missed it, but was there a learning parameter in your model?
No.
Okay.
No, I didn't do any, I didn't model the learning at all.
Okay.
Right, so if you do the V1 lesion, which really is setting a single parameter to 0 in this model, then it predicts that there will still be a dependence of performance on the distractor contrast, because if the correct one also is the most salient, you're gonna pick it above chance. If the wrong one is above the most salient, you're going to systematically choose it and be below chance. But if the two are the same, which is connected by the dotted lines, if they have the same salience, then you should choose left or right 50-50. So it's the beginning of the model, and moving forward, redesigning the experiments, and this is a project that I'm just starting now in collaboration with the Audience Lab at UCSD. We're going to shift to using mice because we would like eventually to dissect the neural circuits through the striatum and the colliculus, which we can do more easily there. We moved the screens to the side of the animal's head, like I have in my model, instead of in front, like in the videos you saw, to simplify, again, the circuitry, so we know that all of the information will be crossed. Putting them in a much larger arena, so they're really running around, so this way, it's this idea that vision for action or vision for movement is a different circuit, so if we want to really probe that circuit, we want to make sure that the animals are moving towards something in space, that's how they're responding. So we may be finding things in this paradigm that would be completely missed, for example, in a head fix preparation, because animals, even though they're just detecting things, those things may not be localized in space, or their response may not represent moving in space to respond to them. And then just switching to something like a Gabor patch instead of these statues and space shuttles, because although they're more boring to look at, they're parameterizable, and we can be more confident that we can match or mismatch those salients. So I just, in the last minute or so, wanted to get back to why I find this such a really interesting question, that we have kind of two brains in our head. We have our primitive lizard brain to offend all the evolutionary comparative anatomists in the audience, right? So we don't have a lizard brain, but we have the tectum and the superior colliculus, which is able to drive our behavior. And we also have the cortex, which is also able to drive our visual behavior. And somehow these two drivers have to get along inside one skull, and every once in a while, one or the other may try to wrest control. And my working model is that the reason we still have a functioning collicular pathway is because sometimes it is adaptive, to override cortex, and you don't want to lose that very long selective pathway. And so there may be some very interesting modulation underlying from moment to moment what is determining the balance of power between between subcortical and cortical control of choices. And to be sure I acknowledge my sources, all of the rat work that I showed today was funded by a grant from KIBM here at UCSD, and the mouse project that I'm just starting was funded by NSF. Anyone who wants to talk to me more about it, there's my email address and my lab's website. And anyone in the audience who is interested in the job, we are hiring a postdoc, an animal technician, not for this project, for a different one. And reach out to me if you're interested in either of those. So I'll stop there and take any more questions. We're kind of doing questions all the time, so.
So at the very end, you said that we have this image of these two pathways. Could there be more? Is this, why, do you think, why two? Yeah.
Yeah, so the retina projects to lots of targets in the brain, but these are the two that have a clear retinotopic map, and so that have the evident substrate for detailed representation of space, but many of them cross the midline. And so in terms of just differentiating left and right space, that could be true for other areas as well. I think these are the top candidates at the moment. We don't have great data on lesioning the colliculus. We tried, and what we found is the animals just didn't do the task, which would be consistent with they just lost the ability to generate an oriented behavior in space. And so then we really couldn't probe anything about what they were seeing. Other people have shown that if you stimulate the colliculus, it biases the outcome. But yeah, so we don't have strong evidence that the colliculus is causing this behavior. And I think the best way to build that evidence would be to record the colliculus and manipulate it.
The colliculus is a more like basal ganglia space, or where?
No,
is it
it's
again?
I don't have an anatomy picture here. So it's underneath the cortex.
Okay. So it's really hard to lesion that one, right?
Yeah, it's big and it's subcortical. Yeah, and it's bilateral. Right. So it's a difficult lesion. And then it's very difficult to work particularly with those animals after it's been lesion. So one can imagine really cool experiments, like we could express channel rhodopsin in retinal ganglion cells and then shine LEDs in the colliculus and signs the terminals. That would be, like, those are the kind of experiments one wants to do, going forward.
Is it possible, but I mean, I guess you would have to know, can we cut out, I guess that's what you mentioned, cut out the connections?
Yeah,
That's not possible.
so I'm not the world's best anatomist, but I don't know, I haven't figured out any surgical procedure that would just disrupt those connections on one or the other. Also, the follicular route to cortex goes through the pulvinar, which is right next to the LGN.
Okay.
And so if you try and knock out one, you have very high risk of hitting the other. So there are some reasons that it's technically tricky. Yeah.
This is kind of tangential, but, my question is, you said that there's like an atopic map that's reserved through at least some of these information transfers from one, nucleus to another nucleus.
Yeah.
But I was wondering, do you ever see, is there, how is 3D space represented? Is there going to be a grid, like a lattice of these cells, or what did you see?
Yeah, so I tend to think of it as a two-dimensional map. Sort of imagine a sphere, an equidistant sphere around your body, and sort of, that's just mapping direction towards which I could be moving, not necessarily encoding how far you would to go in that direction. So there is visual representation of depth, and spatial representation of depth, but I'm not aware of these maps in colliculus in any way of having that depth.
I have a sort of little bit, goes back to your first slide of studies.
Okay.
Evolutionary question. You mentioned that it's equivalent to the optic tecton in fish and frog. And the optic tecton does most of the processing there.
Well, there's no other visual structure. So whatever any visually guided behavior that a fish can do,
Yeah.
it's doing with the structure.
Did V1 like function? Are there some functions that tecton does that diverge into V1 and?
Yeah, so I think the emerging view is that primates are very special among vertebrates in having co-opted the colliculus for eye movements, and in all the other vertebrates, it's more integral to visual behavior. And there are direct cortical targets of the colliculus in or the colliculus through the pulvinar in mice that mediate visual behaviors that depend only on the colliculus, the non-cortex, and those, some are arguing now that that actually was probably the first visual cortex, right? And then the other ones elaborated.
The pulvinar, and then.
Yeah, the collicular pathway was the first pathway to cortex, and then the thalamic one got added on later. All right, well, thanks for your attention. Anyone who's interested, I'd be happy to stay and talk.
Any online questions? Otherwise, please feel free to join us for tea.